on with the chapter on lymphatic and immune systems, and we're going to concentrate on immunity today. There's two aspects of this. There's innate immunity and adaptive immunity. And basically, I want to, in the first half, deal with the innate stuff, and in the second half, deal with the adaptive stuff. The thing is that we really need more time on adaptive stuff. So I'll probably sort of start into that with the at the end of the first half, just to get us in there. As far as the chapter is concerned, the first unit here, well, second unit after the introduction, is about the lymphatic system. And then the next part is the innate immune response. Now, to just break up immunity into the two components, innate and adaptive, innate immunity includes what are sort of separated out here as barrier defenses. Now, innate means to be born with. So this is essentially what you have present in your body at the get-go, ready to go. Kind of redundant there, but and so barriers are obviously, the structural aspect of your body that helps keep things out. The main aspect of that is the skin, which is probably pretty obvious. That's what separates you from the outside world. But there's more than just the skin as barriers. Immunologically, what's outside our body, the way we think of it beyond our skin, is equivalent to what's inside our digestive or respiratory or urinary or reproductive tracts. You could have something and put it in your mouth, and it's not going to make you any sicker than if it was outside of your mouth. So the barrier of the skin is continuous with the mucosal linings of those tracts. And as long as anything doesn't get through any of those barriers, it's immunologically effectively outside of your body. Our conscious perception of inside and outside our body is not exactly in keeping with this immunological definition. And it's not just an immunological definition. Endocrine versus exocrine secretions sort of follow the same thing. Endocrine secretions, like we studied previously, are things that are secreted within your body, meaning really into your bloodstream, and then local signals that stay within a tissue or something like that. And exocrine secretions are things that are secreted outside your body, like sweat or oil or milk or wax across the skin, as well as things that are secreted across these mucous membranes. So digestive juices are exocrine secretions because they're secreted into the digestive tract. And mucus is a exocrine secretion, say, in the respiratory tract, et cetera, et cetera. So the barriers we're talking about here as an immunological definition fit with other sort of definitions as to what's physiologically inside and outside of our body, which is not usually in keeping with what's sort of cognitively inside and outside of our body, I guess is the way to put it. Now, the picture they have here that has to do with barriers to some degree isn't terribly great for everything I want to show, but it does make some good points. We're not going to go into this particular picture in a lot of depth, so I'll just leave it up there in the form that's in the book. And it's sort of concentrating on, well, you can see the nasal cavity and the oral cavity as a couple of examples of spaces that are sort of cognitively inside of our body, but immunologically outside cavity space. And it points out some specializations, especially with the nose. It helps to keep things from getting into our body. But actually, right after this picture, there's a table that lists a few of the barrier defenses. And actually the barriers are more than just the physical structures of the skin and these mucous membranes, but there are a number of active components to them. For example, the skin is a physical barrier in that it has an epidermis, which is stratified squamous epithelium, but then also it secretes a number of things that can be antimicrobial. So in many of the secretions in the in the skin, like sweat and oil and that sorts of things, the pH is a little bit acidic. Bacteria that want to get into our body are looking for an environment that's somewhere in the neighborhood of 7.35 pH. And the secretions in our skin make our skin a little bit acidic comparatively to about 6.5 pH. And that can be enough to inhibit bacterial growth of certain bacterial species. It's not perfect. There are bacteria that are capable of living in a wider range of pH, and so they can exist on the outside of our skin, and when opportunity arises and they can pass through a wound in our skin, then they'll get into our internal pH. And that's how they've adapted to our attempts to limit that with the acidic secretions. Yeah.
So like when you sweat and then like the sweat dry off, when you scratch your your skin and then there's like this dust stuff. Are those like dead bacteria or something like that, or some kind of?
Probably it's more just dirt, skin cells and those sorts of things. You probably wouldn't see necessarily the bacteria themselves. They're probably not, they don't make up enough mass to be visible to the naked eye. And then also in the secretions, there will also be some waste products. It filters your blood, not exactly like the kidney, but still filtering some waste products out. So there'll be some of that mixed in there too. So, there are other secretions that lend a hand to antimicrobial processes. In the saliva, there are enzymes that are released, like lysozyme, which is sort of named for the fact that it is an enzyme that destroys or lyses pathogens. The stomach secretes a very strong acidic solution. Things that can get into our nasal or oral cavities will end up moving along the digestive tract down into the stomach. And most of those things will be destroyed just because of the very acidic nature of the stomach. It mentions mucosal surfaces there. Those epithelial work as barriers also. The last one here is kind of interesting. Most books don't kind of mention this, but I think it's a good thing to kind of pay attention to. What it says there is normal flora. Now the word flora, you probably recognize, it sounds like florist or something like that. And it's usually used to refer to plant matter. But with bacteria, they're not plants, they're not animals, they're very different type of organism. So they're often referred to as flora in the sense that these are the things that live in and on our body. We have a normal relationship with a number of different species of bacteria, some that are living on our skin, some that are living inside our body, like in our large intestine. And in that relationship that we've established with them, not that one day they came up to you and said, hey, can I live on you? But just those organisms and humans and other organisms have developed and adapted within their environment to work together in certain ways. And the flora want us to stay healthy because they've adapted to live in and around our body. And it's best when we're not trying to fight off infections because that can be damaging to them. So they actually contribute a little bit to helping limit potential pathogenic microbes that might get to our system. So they can play a role in that too. So that's sort of what I want to mention about the barrier aspect of it. The rest of the innate immune system or innate immunity is really about the players that help out with this. And there's sort of two aspects of this that I want to address. One is cells that are part of this, and then some chemicals that play a part. Now, one thing about the innate immune response is that it's not specific. And when we get to talking about the adaptive immunity, the point there is that it is specific. And if things kind of go time-wise the way I'm expecting to, in this first half, I'll wrap up the innate immunity and I'll start into adaptive immunity, and I'll talk about that aspect of them being specific and what that means. And then after the break, we'll talk more about adaptive immunity. So the cells that are part of the innate response aren't hunting down specific pathogens. They're just responding to the presence of something that can potentially make us sick. And it's not just about pathogens. It can be about other forms of disease and damage within our system. So there are two classes of cells. The first one is listed here, which is also referring to two types of cells. But they're called phagocytes. Phago, as a root, means to eat. So phagocytes are cells that eat. What they do is they internalize pathogens or cellular debris or whatever might be around and digest that down at the chemical level. That'll destroy the pathogens. It'll clean up if there's damaged tissue somewhere. It'll clean up the debris from that, et cetera, et cetera. Phagocytes are just a class of cells which is largely the various types of white blood cells. Now, there are 5 types of white blood cells. When I was talking about blood, did I introduce you to the mnemonic to remember the 5 types of white blood cells?
Not really.
Does anybody remember the little thing to remember them? What?
Never let monkeys eat bananas.
Yeah, never let monkeys eat bananas. Not really a rule to live by. Your average monkey would like to have some bananas. But those 5 words correspond to the 5 types of white blood cells. N in never corresponds to the N in neutrophil. L in let corresponds to the L in lymphocyte. M corresponds to monocyte. E for eosinophil and B for basophil. And those are the 5 types of white blood cells. That particular mnemonic also tells you them in order based on how numerous they are in your system. So neutrophils are the most numerous. They make up about 2/3 of the white blood cells in your system. And then lymphocytes are the next most numerous, and they make up about 20 to 25% of the cells in your system. And then monocytes come behind that at a little less than 10%. Now, those are ranges, about 2/3, 20 to 25%, less than 10%. But if we add that up, okay, 66 plus 25 plus 10 actually equals 101%. So there are ranges. It's not saying that it is just 2/3 and it's necessarily 25% or necessarily 10%. But those 3 make up the vast majority of white blood cells. The other 2, eosinophils and basophils, make up a very small fraction. I think together they're like 3% or something like that. And eosinophils being slightly more common than basophils. Now, given that eosinophils and basophils are so far behind the others number-wise, we can kind of ignore them for what I'm about to say. So we're really talking about neutrophils, lymphocytes, and monocytes. Lymphocytes are named, obviously, for the lymphatic system. They're important for that. But they are not necessarily phagocytic cells. And I'm saying that in the context of the innate immune response we're talking about here. So if we're going to cut out eosinophils and basophils because they're so rare we don't really want to deal with them. And actually, they're probably occasionally phagocytic too, but we'll just ignore them because they're weird. So we have neutrophils, lymphocytes, and monocytes. Lymphocytes we're ruling out because they're part of something else. They're the basis of the adaptive immune response that we're going to be talking about later on. So that just leaves us with neutrophils and monocytes. Okay? And those are the cells that are phagocytic. Okay? Now, having just said that, you might be staring at the screen going, wait a second, the screen doesn't say monocyte, it says macrophage. The reason for that is that a monocyte is what gets activated to be a macrophage. In the text below that header that says phagocytes, macrophages, and neutrophils, the first paragraph just sort of sets up what phagocytes are. And then the next one, one of the first words there is macrophage, and it's defining what a macrophage is. And then the next paragraph, the last of the full paragraphs there, one of its first words is neutrophil, so it's defining that. And then at the end, the last little itty-bitty paragraph, which is really just one sentence, the bold-faced word at the beginning of that is monocyte. So it just says monocytes are the precursors that become macrophages. Now, neutrophils make up about 2/3 of the cells in your system, and macrophages make up less than 10%. So on average, if we're talking about a phagocyte, it's probably going to be a neutrophil. But macrophages actually tend to get sort of mentioned most of all because they're found in very specific places where we usually think of normal phagocytic activity. There are macrophages in your spleen, which help to clear away dead red blood cells. There are macrophages in your kidneys that do similar things as well as help clear other debris out of your circulatory system. There are macrophages in your the alveoli of your lung that help to clear away any debris that might be airborne that's gotten all the way down to that level and not gotten trapped in the mucus in your lungs. Okay, those are macrophages specifically. So oftentimes you'll hear reference to macrophage instead of just saying phagocyte, because in a lot of situations, the specific places where we see them sort of always active, it is in fact macrophages. Yeah.
What would you say was a relative percentage of lymphocytes again?
20 to 25%. So in tissue like in the lung, in the connective tissue, just hanging out are monocytes waiting to be activated. And macrophages are in the alveoli doing their job. And when a macrophage in the alveolus is sort of clearing away any debris that might have been inhaled or meaning like pollen grains or airborne pathogens, something like that, if the macrophage has sort of gobbled up everything and it's full, like a vacuum cleaner bag is full or something like that, I suppose, then that macrophage leaves and ends up getting destroyed, and a new monocyte comes in, gets activated, and takes up position as the macrophage in that area. Okay, so monocytes are sort of waiting in the wings to become macrophages when they're needed. Okay. Now, I have a video that I want to show you real quick. I've put it on Blackboard in the video content section. And in this section, if you're interested, I only go to some of these. I've embedded a number of videos that I've come across over the years that are relevant to some of the things that we do in class. And they're in here sort of in order. So it starts with the autonomic system, endocrine system, cardiovascular system, and then here's where it starts with the immune system. This is a cute little video that's the ABCs of immunology. So obviously M is for measles, which makes you sick or whatever. If you're interested, you can listen to that. The next one here, however, is actually a little video capture of a phagocyte that's going to be ingesting bacteria. And This is just in a Petri dish. I'm pretty sure those are red blood cells in the background, which look like they're starting to crenulate. So the solution is probably a little bit hypertonic. So here's the macrophage here, and here's a bacterium. And so what we're going to see is the macrophage kind of chasing the thing around until it can finally grab onto it and ingest it. And there's no audio to this. I kind of wish there was a player piano like in a silent movie. We'd kind of go with this. But the thing about these is that they move by extending their the leading edge of their cytoplasm out within the cell membrane, and eventually it makes contact with, there we go, the bacterium and pulls it inside of itself. And it'll be trapped in a little vesicle. That vesicle will merge with another organelle that contains digestive enzymes and has an acidic pH, very much like your stomach. And it'll digest the bacterium that was just gobbled up. And that's how they can get rid of these things. Okay. I have another movie that I'll show you later on that has to do with lymphocytes. That's an even better picture of a cell moving around like that. And I just discovered that other video in the past week or so. So I'm excited to share that this year for the first time. Do you have a question?
And that, so it's via, it's not, what's it? I forgot the organelle that does that. It's
That does
it's
what?
not peroxisome. Right?
No, lysosome. Yeah. Yeah. So the lysosome has digestive enzymes and an acidic pH of about 5 or so. And that merges with the vesicle that the ingested microbe is in. And then much like your stomach, which has a more acidic pH, but still sort of the same approach. It digests down that bacterium until it's destroyed. And actually, there's a picture that I'll show you later on that kind of shows that process and sort of.
Yeah, the upon contact, that was the CD4 and CD8 image.
No. That's different cells. These don't work with that. So that's one of the types of cells that are in the immune response. They're just going to go out, and anything that's in your body that's doesn't look like it's supposed to be there will be cleared away. So that was a bacterium we just saw actually in a Petri dish, but that kind of thing would happen in your body. The way that these cells recognize foreign invaders is because they just don't look the same. The book actually describes it after the next type of cell, which I'll show you in a second, but it's kind of useful to say here. Bacteria, like the one that we just saw there, are not usually motile on their cell, by themselves. Some will have maybe a flagellum or cilia or whatever, they can move around a little bit. But for the most part, the types of bacteria that get into our system are at the whim of their environment, how they move around, which is basically, they can follow water currents or something like that, which sounds kind of strange to talk about inside the body. But as the fluid environment that they're suspended in moves around, they'll move with it. But they don't want to be completely at the whim of those random movements of fluid. So they need a way to kind of hold on to the location where they want to be. And the way that bacteria often do that is in their cell membrane or cell wall, depending on the type of bacterium, they have an array of different carbohydrate-connected molecules. What's a more common name for what we call a carbohydrate? Sugar, right. What happens if you get sugar all over you? Like you've been eating cotton candy. What's the experience going to be like after that? It's going to be all sticky, right? Carbohydrates being very hydrophilic, are basically sticky. And so the outside surface of bacterium is going to sort of interact with its environment in that sort of sticky way to help anchor it so that it's not just, floating around wherever it needs to be. The thing is that those carbohydrates that are on the surface of bacteria are very unlike what we would find on the surface of our cells. Now, some of our cells do in fact have carbohydrates on their surface, but they're not the same molecular structure as the ones in bacteria. So phagocytes can recognize a bacterium very easily. It just doesn't look right. Sort of got the wrong flavor to it or something like that. So we can recognize these things as being foreign. Now, the other type of cell to mention is called a natural killer cell, sometimes referred to as an NK cell. This is actually a lymphocyte. But the lymphocytes we're going to pay most attention to for adaptive immunity are a different brand of lymphocyte, if you will. These are non-specific lymphocytes, and their job is to destroy other cells. Now, the phagocytes are usually sort of picking on smaller cells. So, like we just saw, that lymphocyte, phagocyte there was huge compared to the bacterium it was chasing down. And you might think it's kind of a bully. Natural killer cells basically pick on cells their own size or bigger than them. They usually are targeting body cells that are infected. When a cell is infected with a bacterium or a virus, or when a cell is in some other disease state, it's usually going to look wrong to the cell. Okay? Again, it's not a specific recognition, just this is no longer supposed to be in our body. So phagocytes hunt down and get rid of pathogens and those sorts of things. Natural killer cells will actually get rid of cells that are normally part of our body that have gone wrong in some way. I'm not going to talk about it right now because it'll come up later on, but I want to point it out. In the part of the text underneath the actual killer cell right here, where it's highlighting these two words, those words will come up later on when we're talking about the other lymphocytes. And those are actually the chemicals that they use to destroy other cells. And I'll come back to that when we get to the lymphocytes later on. Now, what I was just talking about with the phagocytes recognizing bacteria because they just don't look right, and then natural killer cells recognizing diseased cells in our body because they don't look right, is more accurately described as the pattern recognition, or no, that process is more properly described as pattern recognition. So there are receptors on these cells that when they interact with the surface of another cell, whether it's a pathogen or one of our body cells that is exhibiting some sort of disease state, the receptor initiates the response that these cells are going to react to. Again, it's not specific like what adaptive immunity is going to be about. Anything that doesn't look right will be sort of taken out by these cells. Okay? So these are the cells that are kind of the players in the innate response. The other aspect of what's going on in the innate response are the chemical players, what here are referred to as soluble mediators. Okay? Soluble just means that it's a molecule that dissolves in the water solution of our body. And then mediator just means these are things that mediate or do certain things within the innate response. The first class of chemicals referred to are cytokines and chemokines. These are types of hormone kind of molecules that are used by white blood cells. So white blood cells usually communicate with each other using these kinds of molecules. And it is true that it's part of the innate response. So if phagocytes are activated to go start hunting down things, they'll start releasing cytokines or chemokines, I can't remember exactly which type, which will help to recruit other phagocytic cells. So they'll come and join in on the fight. Okay. Early induced proteins are a number of chemicals that are usually produced or induced by cells early on in when they're exposed to some sort of pathogenic threat. There's a number of different examples, and I just want to share one, which isn't necessarily what the book is talking about right here, but what the book is talking about to some degree, in the second sentence there's a bold-faced word, which is interferon. And interferons are another type of soluble mediator of innate response. They're a type of early induced protein. There's actually a few different types of interferons, and I just want to tell you the story about one of them. The different types of interferons are just designated by alpha, beta, gamma. There might be a delta, but I can't remember. And so I'm pretty sure it's gamma interferon that I want to mention. Okay, actually it might be beta, I can't remember. But one of these specific ones is part of the response to viral infections. Viruses are unusual compared to the other pathogens that we deal with because the way they work is they infect a host cell, take over that cell, and make it create new viruses. Okay. The way that usually happens is the virus interacts with something on the cell surface, some receptor protein or something else in the membrane of the host cell, and that triggers the virus to sort of inject its genetic material into the host cell. Then that genetic material will be copied and transcribed and translated, and proteins will be made. The copies of the genetic material will then be encapsulated into proteins that are made from the virus's genome, which means that we have new virus particles. The thing is that viral DNA and our native DNA, our human DNA, don't quite chemically look the same. Now, they're made out of the same basic stuff, nucleotides and all that, but we do some little chemical modifications to our DNA to say this is our DNA. It's like Andy writing his name on the bottom of Woody's foot in Toy Story. Like, this is my DNA. Okay. So when foreign DNA gets into the cell, our cell immediately recognizes it not part of our body. It's still going to be used to make new virus particles because not everything can necessarily react to foreign DNA. But something does, and it turns on a gene in the cell which makes this interferon molecule. Interferon's released from the cell into the extracellular fluid of that tissue, which is then going to travel and bind to interferon receptors on the neighboring cells, which tells them that there's a virus in the neighborhood. Okay? It's sort of like if somebody breaks into a house in your neighborhood and the alarm goes off. You know that somebody's breaking into the house because of the alarm, or your neighbor forgot the code again, whatever it is. It's a signal to the neighborhood that there's something there. If any of those neighboring cells have not been invaded by a virus, they'll shut down their own protein synthesis machinery so that the virus will not be able to replicate if it gets into that next cell. That's going to limit the spread of the viral infection. Unfortunately, it's also going to kill the tissue. Now, I shouldn't say kill the tissue. It's going to kill those cells. Oftentimes, that's happening in an epithelium. And so when epithelial cells are destroyed by whatever means, they're usually pretty easily replaced because epithelia grow pretty quickly, pretty easily. So the interferon signal basically tells all the cells in the neighborhood to shut down, possibly at their own expense. They might die off, but the tissue will persist as they're replaced later on. But it removes the chance of any host cells for the virus to infect, and it's hopefully the infection doesn't spread from there. It's sort of a game of chicken between the viruses and your cells. Okay? how long can the virus survive without reproducing, versus how long can the cells survive without making proteins. And oftentimes the cells will die, but hopefully that'll remove any potential hosts for the virus. Yeah.
Figuring out that acts at the level of transcription, or that's not a post-translational modification?
I believe it blocks translation. I think it blocks ribosomal activity in the cell, so the cell stops making proteins. As I understand it, I'm not an expert on that, but I'm pretty sure, because that's where it has to interact with it. Some viruses, their genetic material is RNA, so if you block transcription, that if it's injecting RNA, that RNA might actually be directly translatable. So you really need to get a translation, not a transcription. Then the last of the soluble mediators I want to talk about is this thing called complement. Now, complement, the word here, has a homonym. What does it mean to say that a word has a homonym? Yeah, there are two words that sound exactly the same, but are spelled differently. When you hear the word complement, you probably think of the homonym of this word here. When I say complement, not talking about immunity, what's your first association with that word? What does it mean?
Positive feedback.
Sorry?
The positive feedback.
Well, I would have preferred a less scientific sounding definition, but yes, selling somebody that they look nice or something like that, that's a compliment. That word is spelled Compliment. Okay. Complement without an I, this word here, does not mean, I guess positive feedback is a fair definition, but it sounds too scientific for the point I'm making. Complement in this sense means to work together. Okay. So, and we use that in everyday language too. We can say that
Coffee and sugar complement.
yeah, that's a good example. Thank you. That they work together to make a better, whole thing out of things. And so this is the complement of proteins kind of work together to help destroy pathogens, I guess you could say. And also, I guess it kind of also is referring to the fact that classically they complement part of the adaptive immune response, which is antibodies. And we'll get to the antibodies in a second here. The complement proteins are referred to simply, by designation C for complement, and then a number after that. So C1, C2, C3, C4, et cetera, et cetera. There are up to 9, C9 at the end of this. Now to describe what happens, I actually want to work backwards through this picture. Down at the bottom, we have a pathogen. This green lumpy thing is supposed to be a bacterium, maybe. And complement acts as complement forms a channel-like structure called the membrane attack complex, which is basically a big channel made up of a bunch of little individual proteins, many of which are complement. So there's C5, C6, C7, C8, and C9 that are all part of the membrane attack complex. But there are some other ones too. So there's different color coding in here suggesting the different particular complement proteins. Now in this picture, this membrane attack complex that's demonstrated here is actually very out of scale. It's not going to be that big relative to a bacterium. We're just trying to show you kind of what it looks like. But these other things around on the bacterium are supposed to represent other membrane attack complexes. So what it is it makes a hole in the membrane of the pathogen, and the insides leak out, which is going to cause the pathogen to be destroyed. Okay. That's the end game for complement. the way that the membrane attack complex forms is that this component here called C5a is going to recruit C6, 7, 8, 9 to form the complex. C5a comes from C5, and the A designation is just talking about it being part of that molecule. The other part is C5b. C5 is broken in half, or broken into these two components, by another part of the system called C3b. And again, the B designation after it is referring to it as being part of the overall C3 protein. So C3 is broken into two parts, and one of those parts breaks C5 into two parts, and then one of those parts recruits the rest of the proteins to form the membrane attack complex, and the pathogens destroyed. That's essentially what complement's doing. How we get to this point, there's actually two different ways that it happens, and only one of them is shown in this picture here. The one that's shown in this picture involves antibodies, which are represented as these blue things here. And antibodies are made by cells of the adaptive immune system. They're specific recognition markers for whatever this invading pathogen is. So if this is an E. coli bacterium or something like that that you've ingested, you'll have antibodies against that will bind to the surface of the pathogen and initiate what's going on here. That aspect of it, the antibody binding to it, is not an innate immune response. That's part of the adaptive immune response. But the rest of it is complement. With that specific binding, the antibodies will interact with C1, which we see here, which will then take C2 and C4 and break them down, and pieces of those two come together to form what's called C3 convertase, which will then break down C3. Okay. This is referred to as the classical complement pathway. It was the first way it was described. It's dependent on antibodies. But later on, it was discovered that complement can work in a different way also, a non-specific way. Without antibodies present, there's another way to get to the point where C3 is broken down. And that's referred to as the alternate pathway. So complement has two pathways. There's the classical pathway, which interacts with antibodies, involves C1, C2, and C4. And then there's the alternate pathway, which does not involve, or does not, interact with antibodies, so it's non-specific. And starts with C3, which goes to C5 and C6, 7, 8, and 9 to form the membrane attack complex. Let me put the C's in here just so that we can keep in mind that those are also complement pathways. So what I started out talking about was really the alternative pathway, and then I added to that what leads into that as the classical pathway. And we see that demonstrated here. Now, I want to talk specifically about something else in here that's not mentioned, that I haven't mentioned yet, which is what happens with the other half of C3. So C3b goes on to help initiate formation of the membrane attack complex. C3A does something else. The way it's depicted here, it's a little oversimplified. Here it just says that C3A attracts phagocytes. Because this is going to be happening where there's an infection present, and so the concentration of C3A in that area will attract phagocytes to the infection site. But I want to expand a little bit more on that, on how it does that. What C3A does is it actually activates mast cells. And what mast cells do is they release histamine. Histamine causes the inflammatory response, which I'm going to talk about in a second. And from that, phagocytes will be mobilized and come to that region. And then C3, I can't remember if it's A or B exactly, interacts with phagocytes, and they know what they need to phagocytose because of the higher concentration of these things in the area. Okay. So it's not just that they attract phagocytes, they actually do it by interacting with the mast cells and initiating inflammation. Okay. So there is a little bit more to what complement can do rather than just killing the pathogen itself. It's also going to help bring in the cleanup crew to get rid of that. Okay. Which then brings us to considering the inflammatory response. Now, the inflammatory response is essentially what we think of as swelling, okay? But it's more than just swelling. It is an increased perfusion to the tissue. By perfusion, I specifically mean blood flow. So in this picture, we see a wound, and actually, let me make this picture a little bigger. Okay, so there's a wound here. This is supposed to be the skin, and the little wavy line here represents the basal lamina between the epidermis and the dermis. So the wound has gone all the way through the epidermis, which means that that barrier is no longer an effective immunological barrier. And so bacteria can get in through that cut. The damaged tissue there will start to release some signals, as damaged tissue will, which will initiate the inflammatory response. It does that through mast cells releasing histamine. Histamine, when it binds to receptors on blood vessel walls, causes dilation of the blood vessel, and which is not, and the second thing, which is not represented in this picture, is it also increases the permeability of the blood vessel wall, which means that it makes the blood vessel leakier. So stuff in the blood can get out of the blood easier. And so some of what's in the blood that's going to get out easier is water. And so the tissue is going to swell. And in the second picture, they kind of depict that. Okay? So here, where the wound is initially, the skin's relatively flat. And then it starts to bulge out because there's an accumulation of fluid in the neighborhood. So that's the initiation of the inflammatory response. Now, inflammation is actually referring to the sort of lighting on fire of the tissue. Flame and inflammation referring to fire. So it's not just about swelling. As the blood perfusion increases in the tissue is going to get warmer. Now, the bacteria that are getting in here are comfortable at a temperature of about 37 degrees. So as the tissue gets warmer, that will help to inhibit microbial growth. More than just the blood showing up there and making it a little bit warmer, the tissue will also potentially get warmer because there are chemicals released called pyrogens, which increase mitochondrial activity and therefore increase the temperature of the tissue. Okay. Now, that can happen locally, which is part of the inflammatory response, but it can happen systemically too, which is what we call a fever. And so as your systemic temperature goes up, you're going to hopefully inhibit microbial growth, which will get rid of an infection also. It's just that can get very dangerous. Systemic fever, if it lasts too long, can start to damage tissues in your body too. Now, between these two pictures, we see a white blood cell or a couple of white blood cells here in circulation, and then we see a couple of white blood cells off to the side here. What happens with that increased perfusion, and especially with the permeability of the blood vessel wall, is that white blood cells, mainly neutrophils, can move out into the surrounding tissue. They'll migrate across the blood vessel wall, and they'll become phagocytes. Now, the way that they can do that actually has a lot to do with the cellular structure. Okay. In this picture, the two white blood cells they show, they've drawn their nuclei in the kind of characteristic way that we see for white blood cells. Instead of being a spherical nucleus, it's usually sort of in connected little lobules of the nucleus. This one down here is maybe a little bit more dramatic example of that. So here's a chunk of the nucleus and a very thin nuclear bridge connecting to the next part, all that sort of thing. This makes it possible for these cells to squeeze between intracellular clefts and get into the surrounding tissue. It's a lot easier for the cell to push a little bit of its nucleus through at a time rather than trying to squeeze this big spherical nucleus through all at once. It's kind of like, you've probably seen these kind of things on the internet plenty, cats trying to squeeze their way into weird little places. Okay. They're very flexible, and they can get into very small places because they can just squeeze a little bit of their body through at a time. And it's actually, kind of fun to watch on the internet there a little bit. But that's what's happening here. These cells are going to squeeze through a little gap in the wall by pushing the cell through with a little bit of its nucleus, and then a little bit more of its nucleus will follow, and a little bit more. And it'll be able to just slip through and get into the surrounding tissue and act as a phagocyte when it gets there. Okay. And here we see one of those phagocytes is engulfing this particular microbe, and it'll destroy it. Now, of course, in the video we saw a little bit ago, the cells don't maintain a constant shape. They keep moving. But for the sake of making the drawing here, the artist just copied and pasted the white blood cell and apparently put a green thing on top of it. That's phagocytosis, but that's what's happening there. Now, a couple of other things that happen with the inflammatory response, which are not really clear from what's going on here. With damaged tissue, there's going to be pain associated with that. So if you cut your skin like this, it's going to sting a little bit. Why that happens is because the damaged cells release chemicals that activate pain fibers. And we actually talked about that a little bit back when we were talking about hormones. And I was describing the eicosanoid hormones, which include prostaglandins. Prostaglandins are released by damaged tissue as a hormone or paracrine signal into the surrounding tissue so that your pain fibers are activated. And it's more of a behavioral cue. It lets you know that damage has occurred so that you can respond to that appropriately. So if you cut your finger like this, you might pull it away from whatever might have caused the damage. If you step on something, if you step on a task or something like that, that pain is going to cause a reflex where you pull your foot away from whatever you stepped on. So that the pain is sort of there to stimulate some sort of behavioral response. Sometimes it's reflexive, sometimes it's a little bit more complex. If you sprain your ankle, you're going to have an inflammatory response there, and the pain that you feel in the sprained ankle is probably going to make you put more weight on the other ankle, which will help that damaged ankle heal a little faster, because you're not going to ask it to do as much work. Also with the inflammatory response, you'll see a loss of function in the tissue. So the sprained ankle example is pretty good. With the swelling around a joint, the joint's going to lose some of its range of motion. Okay? So the joint has lost some of its function, and it's going to be able to heal a little bit as you're not asking it to do as much as it does. But the same thing would be true if you think about other inflammatory responses. In any condition that ends in itis, which means swelling of whatever, there's going to be some loss of function. Okay? So meningitis is an infection of the meninges around the central nervous system, and that's going to compromise nervous system function. Okay? Or pancreatitis is going to result in the loss of some pancreatic function, more digestive than insulin, that kind of thing. But that's just because that's more of what the pancreas actually does. So there's going to be some loss of function there to some degree because of the inflammation. Okay? So this is sort of the last thing that's covered in the innate immune response stuff, but to some degree, it's the beginning of the innate immune response. So if you damage the tissue like we see here, that's the beginning of the infection. And with that damage, immediately the inflammatory response starts to occur. In the book, they put it off to the end, I think, because it sort of follows after talking about these chemical or soluble mediators, because a lot of the inflammatory response is based on chemicals that are released from damaged tissue or the histamine that's released from mast cells and those sorts of things. Sort of follows after that part here. But really, it's the first thing that happens. There's going to be an initial chemical response within the tissue, which is going to stimulate the inflammatory response right off the bat. And then everything else will kind of come in behind that. Okay. Now, okay, so that sort of brings us to the end of that section of the book. The next two sections, here and here, are talking about the adaptive immunity, and it's segregated into the two main types of lymphocytes that we want to consider, which are T cells and B cells. But really, to set up what the adaptive immunity is about, I want to talk a little bit about something from both of those. So let me do that, and then we'll take a break, and then we'll continue on with this. So let me grab a picture out of the T cell section and another picture out of the, oops, too far, B cell picture section. Okay. This is what's called a T cell receptor, or it's abbreviated TCR. This represents an antibody, which you've probably heard of before. Antibodies are made by B cells. T cell receptors obviously are receptors on T cells. They are essentially similar material. No, they are similar components to the two cells that are the basis of the specific responses that we see in the adaptive immune response. So I want to sort of describe it to begin with the antibody. So an antibody is actually made up of two mirror image structures. There's a shorter piece of protein called the light chain and a longer piece called the heavy chain. And there's two of each of those. So the yellow and green things are both light chains, and the blue and red things are both heavy chains. The proteins take on a configuration that schematically looks kind of like this Y, and off to the side, this is a little bit more specific or actual protein structure representation. And the colors are the same, so the yellow and the green and the blue and the red are the different chains there. Where the two heavy chains come together down at the what I like to call the stem, not that that's anybody else's term for it, where they come together here, and then where they're each connected up to their individual light chains, there's some very thin sections of the protein which are actually very flexible. Okay? They're like hinges. And so these two pieces here don't have to be in that particular Y-shaped configuration. They can kind of go wherever they need to interact with whatever they need to interact with. In that Y section on either side, where the light chain and the heavy chain come together, there's a region in the light chain and the heavy chain each called the variable region, and then the rest of it's constant, as is the rest of the heavy chains down below. What this is referring to is just genetically, there's a highly variable part of the gene that codes for this part out here, and then the rest of the gene is constant or very stable. Within each lymphocyte, B lymphocyte, when it's first generated, the gene it has for antibodies goes through a little randomization of the variable region. Little chunks of DNA are cut out and reinserted randomly in different order. Okay, this happens for all of the lymphocytes that are made. Each makes its own little unique antibody gene. And so it's going to make its own little unique antibody. And what's going to be unique is out in the variable region at the end, what's represented by this kind of weird scallop shape is a unique three-dimensional shape to the protein, which will then be able to bind to a particular shape of an antigen. And that antigen can be a molecule or a pathogen or all sorts of different things. Just whatever it is, it's going to have a three-dimensional shape, and it's going to bind to this specifically like two puzzle pieces meeting together. Now, the light chain and the heavy chain of an antibody are just duplicated, and there's two of each. So on the other side, the antigen binding site over here is exactly the same. Now, in the picture, they actually draw it a little bit off. So if you look at how it's drawn over here and how it's drawn over here, they just inverted one of these things, so it's backwards. But really, it should be identical on either side. It can bind to two copies of the exact same thing. Now, if we go over here to the T cell receptor, they're very similar in their structure. There's an alpha chain and a beta chain rather than a light chain and a heavy chain, because the alpha and beta chains are the same size, same weight. So we just distinguish them differently. There's only one set. It's not duplicated like in the antibody. So there's just one. There's a variable region, which is the outermost part of the receptor, and a constant region and a transmembrane region. Really, the transmembrane region is constant also, but it's specifically named because it's what goes through the membrane to connect the receptor to the cell. So the constant region and transmembrane region here are very similar to the antibody structure here. Okay, so we have a variable region and constant region, variable region, constant region. It's the same basic structure there. In the variable region of the antibody, like I just described, it rearranges its DNA so that it makes up a unique three-dimensional structure at the antigen binding site. The variable region of the T cell receptor does the exact same thing. In this picture, they just haven't depicted it with the same sort of scalloped pattern to suggest a unique shape there, but that is true. the T cell receptor is going to bind to antigens also. And oops, going back with sorry. Going back to the T cell part, right after the T cell receptor picture, there's this other picture here that shows 3 different T lymphocytes, each one expressing their unique T cell receptor. And they're indicated as unique because the variable region in each one is colored differently for the sake of this picture. So this particular variable region binds to this particular piece of the protein that we're looking at. This one binds to a different piece. This one binds to a different piece. Where they each bind is referred to as an antigenic determinant. That's the 3-dimensional shape of the antigen that fits like a puzzle piece into the antigen binding site in the variable region of the T cell receptor. Okay. We can have a picture just like this with 3 different antibodies and showing it binding to 3 different places. They just happen to depict it with T cell receptors here. Okay. But the same sort of things happening in antibodies. That's the basis of the specificity that I've been talking about for the adaptive immunity. It's adaptive because each of these cells is making a unique T cell receptor or antibody. And after birth, in the first 6 months or so of life, this part of the immune system adapts to the environment. You weed out the lymphocytes that make T cell receptors or antibodies that would make you sick by reacting with things in your body. And you keep the ones that could potentially protect you against things that might invade your body in the future. So there's a little time at the very beginning of life where your innate immunity is present because you're born with that. And your adaptive immunity has to take a little bit of time to adapt to your particular environment. And then once the adaptive immunity is online, which happens within the first couple of years of life, then your immune system is going to be able to be fully functional. And it'll persist and be ready to deal with anything that you might interact with throughout your whole life.
Doesn't say like the thymus that it matures until adulthood?
We're going to talk about the thymus in more detail as we explain what's happening with these after the break. So, yeah, in adulthood, the thymus is sort of done doing its job. Your thymus, my thymus, most everybody's thymus in here is just fat. After it finishes doing its job in adolescence, it just becomes adipose tissue. And we don't see it in a model like this because in an adult, which is what this model shows us, it's a non-functional gland. Do you have a question? Or you just So this sort of sets up the idea that this is about specific reactions. What I want to talk about is sort of how the T cells and the B cells come to mature and be ready to do this, and then a little bit about how they react to antigens and do their job. This is a nice spot to stop so we can take a break, and then we'll come back and do this. It's a little earlier than I normally would break probably, but so we'll take about 20 minutes. Be back in here by the clock up there at, what is that, 2:30? Yeah, 2:30. And then we'll continue on and I'll finish up and we'll talk about the adaptive immunity for the rest of the time.